Hello everyone. Today we are going to talk about Mendel's monohybrid cross. Genetics is the study of genes, variation, and heredity. Mendel was an Australian monk and he worked with genetics and inheritance of traits. Mendel investigated inheritance of 7 different features in peas, including height, flower color, seed color, and shape. In a monastery, he became inspired to begin his research on heredity, which would make him the father of genetics. Mendel chose pea plants because of a variety of reasons. Pea plants have a small life cycle. They are bisexual and can be self fertilized or cross fertilized in a controlled manner. They can also produce a lot of seeds. But most of all, he chose pea plants as they had a variety of contrasting characters from stem, pod, seed, and flower. So, stem size could be long and short, and the flowers on the stem could be axial or terminal. The pod form could be full or constricted, or the color could be yellow and green. The flower color could be white or violet. The seeds could have the cotyledons or the form as a variation. For his experiment, Mendel chose true breeding plants which were tall and short plants, which meant that these plants on self fertilization bore seeds which only yielded offspring of the same size as the parents. So, tall true breeding plants produced only tall plants on self pollination, and short true breeding plants only produce short plants on self pollination, generation after generation. Now, let us look at the experiment he performed. For parent generation, he took tall and short true breeding plants and made them cross pollinate. That meant that the pollen from, say, the tall plant was dusted on the stigma of, say, the short plant. From this cross pollination, the seeds were sown, and all the offspring from the tall and the short plant grew up to be tall plants. Now, the next generation, or the F2 generation, led to results which confused Mendel altogether. So, the progeny that was obtained in the F1 generation, Mendel decided to self fertilize them. The first generation tall progeny yielded a very different result. This time, there were short plants. What Mendel has as a result was very different than what he had predicted. So he crossed a tall plant with a dwarf plant and observed how traits are transmitted in the progeny. He also calculated the percentage of tallness or shortness in subsequent generations. In the first generation, the F1 or the first filial generation, Mendel did a cross pollination between the tall true breeding plant and the short true breeding pea plant. He got all pea plants in the first generation which were tall. So F1 yielded all tall progeny. But in the F2 generation, he made his F1 generation undergo self fertilization and he identified a constant ratio in which the traits presented themselves. So he observed that in F2 generation there was presence of shortness, and the tall plants and the short plants always presented themselves in a fixed ratio of 3:1. What Mendel got as a result was very different than what he had predicted. According to the blending model, both the traits of the father and the mother should have gotten blended, and the progeny should have been somewhere in the middle of those traits. So, if there was a tall and a short plant, they may be expected all the progeny obtained to be medium sized plants. But actually, they weren't. So, Mendel hypothesized the following. Mendel hypothesized that each character is controlled by a pair of factors. In the first generation, all plants are tall and shortness trait is not expressed. Such traits which express themselves in the presence of another trait. So here, since tallness was able to express itself better in the presence of shortness being present, or short plant or short parent being present, we call tall as the dominant trait because tall is being expressed prolifically. But on the other hand, we have shortness or short plant, the trait which is unable to express itself in the presence of, say, tall plant or the dominant trait. This type of trait which is unable to express itself in the presence of a dominant trait is called a recessive trait. This is a representation of a monohybrid cross. So we took two parents, one tall true breeding parent which will have a pair of alleles represented by capital T, and the dwarf plant will also have a pair of alleles represented by small t. So when both the alleles, like in the true breeding plants, are same, this is known as the homozygous conditions. What are alleles, you may say? Alleles are basically two variants of a gene. So we are diploid organisms because we have two alleles, one inherited from each parent. During the time of gamete formation, only one is passed from each parent. So here, the tall plant will only pass one gamete, and the dwarf plant will also pass one gamete. And when the fertilization occurs, the gametes fertilize and restore the number of alleles. So now we have one allele from the mother and one from the father. So here you have capital T from the tall parent and small t from the short parent. In the F1 generation, when we fertilize, we get the genetic makeup as capital T small t, which has resulted in a tall plant. The genetic makeup is what we call the genotype of the progeny in the F1 generation. So what is the genotype of the progeny? It is capital T small t. But the phenotype, or what is the expression or the physical expression of this genetic makeup, is tallness. So even though it has a short t present from the small plant or the dwarf plant, we still will have the physical expression as tallness. As in the Mendel's experiment, the F1 progeny was self-pollinated to receive the F2 generation. So here we have two parents, both are tall physically, but their genetic makeup or the genotype contains an allele from the short parents as well. So the tall plant has the genetic makeup of capital T small t, and another tall plant, because it is being selfed or it is being self-pollinated, we have the same genotype. Here the gametes that will be obtained will be of two types. capital T, small t from each of the two parents. So each of these gametes from a parent can at random fertilize with any one of the two gametes from the other parent. To know all the possible recombinations of gametes, we can draw this checkerboard, also known as a Punnett square. So we can see through this square what will be the genetic makeup of the progeny. Inside we have as progeny capital T, capital T, capital T, small t, capital T, small t, and small t, small t. These four are the possible recombinations of the progeny then we can get. So beginning from the first capital T, which is homozygous, will yield a tall plant. Then the second one, which is capital T, small t, is a heterozygous condition. That is, both the alleles here are now different. So here, in the presence of the dominant trait, the smallness will not be able to express itself. So capital T, small t, even though it has the trait from the small parent, will also yield a tall looking plant. So the genotype will be capital T, small t. It will be a heterozygous condition which will yield a phenotype or the physical characteristics of a tall plant. But now in this case, we also have a small t, small t. So here there is no dominant trait present. In this case, the recessive trait will now be able to express itself. And the phenotype of this plant will now be short. So let us count now. In the F2 generation, second filial generation, we have 3 tall plants and 1 short plant. This is known as the phenotypic ratio. While the genotypic ratio or the genetic makeup is 1:2:1. 1 is the homozygous tall plant, capital T, capital T. 2 are the heterozygous tall plants, capital T, small t. And 1 is the homozygous short plant. Now let us quickly look at some terms to familiarize ourselves. First is phenotype, which is the outward appearance of a character in an organism. The genotype is the genetic makeup of an organism. What is the dominant trait? The trait which expresses itself even if there is another trait present, like tallness or capital T, capital T. Recessive is the gene which is unable to express itself in the presence of a dominant trait. It generally expresses itself in the homozygous state. So when there is no presence of a dominant trait, like in small t, the recessive trait is able to express itself. What is the homozygous condition? Where both of the alleles are the same, like capital T and small t. Heterozygous is when the two alleles at a given point are different. So capital T, small t together is a heterozygous condition. Now at the end, I have two questions for you which you can try on your own. This will build an understanding of what you've just learned. I hope you understood this well. Thank you all.